in this mutations lesson that I wanted to go over with you. So just starting off, I'm sure we've all heard of mutations before, especially in grade 10 when you guys talked about cancer and sort of problems in the cell cycle. Um, but just to kind of go over briefly, a mutation is a change to the DNA sequence. And what I mean by that is the A, T, C, and Gs, those nucleotides that we talked about before. And we've talked about the fact there are 3 billion of those in our genome. So there's lots and lots of those base pairs. But unfortunately, if even one of them is wrong, that can cause some problems. Now, unlike last year, where we talked about only negative types of mutations, it's possible that actually a mutation is beneficial or helpful. We're going to talk more about that when we get into the evolution unit. But just to really briefly mention is they can be induced or random. So induced meaning that we have sort of like a known cause. So things like UV radiation, certain chemicals are known to do this. Some viruses can cause mutations and so forth. The list goes on and on. But unfortunately, some can also be random. So we can't necessarily predict them. And actually, even just in the copying of the DNA, this is possible to happen. It happens pretty infrequently, the random type, but it's very possible. Okay, so not caused by like in any external sort of sources. Now, last year you talked about interphase a lot with the cell cycle. During interphase, we've talked about even in the genetics unit, the cell is checking for mistakes. And so if it finds there is a mistake that it cannot fix, it will self-destruct, which we call apoptosis, which is right here. And we also kind of call that a programmed cell death. So again, this is if, or in the case, I should say, that there is a problem that cannot be solved by the cell. It doesn't want to keep passing that along. So it will try to self-destruct. So I want to go over really briefly is the two types of mutations. We have a point mutation, a mistake in a single base. So I was talking before about the A, T, Cs, and Gs. So on this side, we have the normal sequence. It should be ACA. But there's a few different things that can go awry. First of all, we could have what's called the substitution. Instead of a C, they've put a G in place. Again, this could be induced or random. Likewise, insertion, we have an extra whole base here. So a sequence that should be 3 has become 4 now, which is not good. And likewise, deletion, we should have 3 here, but we only have 2. Okay, so we call these point mutations because it's a mistake in a single base. So in this case, the C is substituted out or something extra is inserted or so forth. The second one are much larger scale. So more than one base changed at a time. And we've actually talked about some of these before, which is like a monosomy or trisomy. Those are the non-disjunction disorders. But I want to go into some that are a little bit newer, which are other sort of examples. So changes to the chromosome structure. So I'm not talking about here a whole extra or a whole fewer chromosome like monosomy or a trisomy. I'm talking about sections of chromosomes that have completely moved to somewhere else. So here I have a normal chromosome 1 and a normal chromosome 7. But a mutation happens that causes a whole big piece of chromosome number 1 to join chromosome number 7. And I mentioned here translocation, which is what we call this mutation, is not the same as crossing over. Crossing over is where they exchange genetic information, but they have to be homologous. In other words, chromosome number 1 switches information with chromosome number 1, right? They're the same type of chromosome, they have the same information. In this situation, a whole big piece has just moved to a different location. And that's really, really dangerous, actually. We're going to talk about at the end of this lesson if big sections are incorrect, that can cause problems with the protein that gets made. And you focus a lot more on that in grade 12, but I'll just intro it to you today. Then down here, we have a couple more examples. And so what I'm drawing here, this would be, again, a normal sort of chromosome. And here I have sections 1, 2, and 3. So these could be genes, for example. Well, in the sort of copying of this DNA, or it could be also induced by something else, at some point there is a deletion. So one of the genes in one of the sections is totally removed. So here we have like our mutated copy. What should be there, which is number 2, is actually disappeared. Okay. And in C, we have sort of the opposite problem, duplication. I should have gene 1, 2, and 3 on this chromosome, but now I've actually duplicated one of those sections. So that's called a duplication. And I mentioned here that if it's repeated 3 or more times, that's a particularly dangerous situation that can cause lots and lots of issues. So we have duplication of a section. So it doesn't even have to be a whole gene. It could be just a small sequence that gets copied many, many times. So again, can cause different disorders and so forth. And last but not least here, is inversion. So inversion is where we have basically a reversal. So where it should be in order 1, 2, and 3, we now have it inverted 3, 2, and 1. Things have switched places on the same chromosome. I've also shown it another way over here. I have those same genes are still present, but they've been moved to the wrong location. And again, these are pretty dangerous because what we see happen is the information is in the wrong place. And whenever that happens, you're going to have issues with the protein that's ultimately made. Okay, so a little bit more about inheriting mutations, and we spoke about this when we dealt with non-disjunction and meiosis, so way earlier in the unit. Here, what I'm describing for you is that mutations to body cells are not heritable. Okay, only mutations in gametes are heritable. That means that only those mutations that affect the gametes can actually be passed on to our offspring. And the example that I give for this is skin cancer. So if I develop a skin cancer, okay, let's say on my hand, the skin on my hand, that becomes mutated by UV radiation and it develops into skin cancer. That skin cancer is not actually making its way to my gamete cells. And so if I were to have children, that particular mutation is not going to be passed on. Okay, only things that happen to the gametes are going to be passed along. Okay, so that brings us to the type of mutations, and we're going to talk a lot more about this in lesson 1 of evolution, which you're going to do later this week. And a couple examples I'm just going to show is that mutations are not always a bad thing. They can be neutral or beneficial as well. A really good example of that is sickle cell anemia. I'll talk more about that in lesson 1, which is the red blood cells carry oxygen to our body. We know the sickle cells are misshapen, and so they can't do so as efficiently. They also have other issues such as easier to clot and cause blockages and so forth. But in places where malaria is present, so countries that have high incidence of malaria, this sickle cell is actually impossible for malaria protists to actually infect. So the people that have this are actually immune to the disease. So in that particular environment, this normal red blood cell is actually not as beneficial to you as having a sickle cell. So that would be a great example of location-based evolution. So places that are prominent or where malaria is prominent, I should say, are going to have more of this sickle cell. And I will talk more about that later on, which is in lesson 1. And another example, lactose tolerance and intolerance. So what you may or may not know is that actually most people are not naturally able to digest lactose. The percentage or the frequency I found was 75% of adults worldwide are actually lactose intolerant. But some people have a beneficial mutation which allows them to digest it as they age. So after the point of breastfeeding in infancy, some people are able to digest it just fine. They actually have a mutation for that. And then the last example here, which I'm not going to speak too much about because we talk a lot more in lesson 1 of evolution for this, is antibiotic resistance for bacteria. So it is a very beneficial mutation for the bacteria, but it's very harmful to us, as we also discussed in the first unit. And this is all due to a random change, a random mutation to the bacteria, which allows it to survive treatment by antibiotics. So the one that I really want to explain to you guys is on the last page. So I want to just explain to you why I've included this here. This is actually a concept that you talk a lot more about in grade 12. So I'm not going to go into too many details about this, but what I want to show here is basically our DNA sequence. And the DNA sequence is the instructions. And I've mentioned this before. What is it instructing to do? Okay. The goal of the DNA is to essentially give instructions to make proteins. Okay. And so it goes through many, many steps, which is represented by these arrows in the middle. And I've mentioned down here, proteins have important jobs in cells, including making our phenotypes. So in our genetics unit, we've talked about lots of different phenotypes that can happen. I'm going to use just a really simplified example. Eye color. Okay, we all have different colors in our eyes, and that is caused by our DNA's instructions to make pigment proteins. So that's what gives things its color. So over many, many steps, which we're going to discuss in grade 12, the DNA essentially is made into those different proteins. So on the sidelines here, I've written central dogma, and that is basically the main principle of biology, which tells us how do we get from DNA all the way to a protein that has some function, okay? And I've mentioned just here is essentially what we call those steps. You're not going to have to know that for anything this year in grade 11, but it's just a basic sort of outline that we take information from DNA and we eventually make that information into a protein in the cytoplasm. And as I mentioned, that protein will have tons of jobs. There's millions of type of proteins that can actually form, all of which have its own specific function. So I want to just draw upon this example to talk about sickle cell anemia. So here I've put a normal DNA sequence. So you can see on the side, this is DNA. So where I should have GAG as a DNA sequence, people with sickle cell disease have GTC. So they have one single base pair that is mutated. Okay, so thinking about whether that is a point mutation, substitution, deletion, insertion, and so forth. So what that means is that in the normal phenotype, which is GAG, they're going to make a protein sequence that looks like this: pro, glue, and glue. These are amino acids. I'm going to speak more about them in Unit 4. But this sequence is going to make a certain protein, which is eventually going to form our red blood cell. So this would be our normal red blood cell, our normal phenotype. In the mutated sequence, I don't have pro, glue, and glue anymore. Now I've changed the protein, pro, val, and glue. And what happens here, this valine that's now inserted here, which shouldn't be, it's going to cause a slight change in the shape of the protein. And it might not seem like a big change, like only one of these subunits or one of these boxes has been changed. But in fact, it changes the entire shape of the protein. And if the cell makes lots of copies of this, which it will do, you're going to have lots and lots of slightly the wrong protein. And what this eventually forms is the sickle cell shape. So it's not in the shape that it should normally be, okay? It's misshapen. And the problem that becomes with that is that if all of your red blood cells are like this, that's going to cause issues like clotting and so forth that I was mentioning before. So again, this last page, it's really going into concepts from grade 12. I just introduced it for you so to make sense of the fact of why is just one single thing really a problem. It's that it makes the incorrect proteins. And I'll leave it on that note for now. We'll continue that conversation in grade 12 if you take biology next year as well.